everybody, and I hope I'm audible to the audience. Yeah. So good evening, everybody. First of all, I would like to thank everyone in the audience for joining us for this session of webinar, especially on a Friday night, India time. So this is a webinar, CCMB Biologue is a webinar series that we started in July 2020, right in the middle of pandemic. So before if the lockdowns happened, we used to invite various speakers to CCMB to give talks. And since that was not happening anymore, and more of the academic community was switching to a online mode of function, we decided to make use of this opportunity. And then we decided to contact experts of life sciences in different fields of life sciences across the globe. And we started inviting them to give talks at CCMB. So since July 2020, we've had talks by around 16 people from different areas of life sciences, such as neurobiology, then structural biology, biophysics, ecology and evolutionary biology, and then even some science communicators. And this entire Biologue series is coordinated mainly by PhD students in our institutes, Center for Cellular and Molecular Biology. And our audience also generally includes PhD students in life sciences, as well as a certain amount of undergrads and maybe postgraduates, etc. And this talk today is going to be around 40 to 45 minutes long, with a 10-minute question and answer session followed. So I'd like to request Professor Jyotsna Dhawan to introduce the speaker and moderate the talk, please. Thank you.
So thank you to all the CCMB students for this lovely Biologue series. It's kept us going through the pandemic. It's always exciting to have the students bring on new and exciting speakers. So, and also thank you for the opportunity to introduce Fiona Watt. Fiona is internationally known for her work on stem cells in both healthy and diseased tissue, in skin specifically. And, you know, Fiona took her DPhil from Oxford and then was a postdoc in Howard Green's lab at MIT in Cambridge. And she started her independent career at the Kennedy Institute for Rheumatology in London, and then moved to the London Research Institute. She has been the Deputy Director of the Cancer Research UK, Cambridge Research Institute, and Deputy Director of the Wellcome Trust Centre for Stem Cell Research at Cambridge. In 2012, she moved to her current position as the Director of the Centre for Stem Cells and Regenerative Medicine at King's College in London. And in addition, she's the Executive Chair of the MRC in the UK. And as if she didn't have enough to do, she's now the new Director of the EMBO. So Fiona, I should tell you, I first heard about you when I was a graduate student in the 80s in Boston, because postdocs from Howard Green's lab were creating waves in the cell biology of differentiation. Elaine Fuchs and Fiona Watt were studying epithelial cells, and Bruce Spiegelman was studying fat cells. And this cohort has really stayed at the forefront of differentiation and stem cell biology, defining, I think, the importance of basic cell biology in understanding disease mechanisms. And Fiona's new work with single cell transcriptomics is a great example of that. So it's my great pleasure to ask Fiona to speak to us about her work on cell heterogeneity in epithelia. Over to you, Fiona.
Thank you very much for the kind introduction, and a huge congratulations to the PhD students and postdocs for pulling this series together. I think all over the world we've seen the power of the new generation of scientists, and I don't know what the future holds in terms of the way we're going to interact post-pandemic, but we've certainly learned a lot, and I'd like to pay tribute to you for that. So I'm now going to share my screen, and me just do this. Can I, let me just, sorry, I'm on two screens. Let me go with that. And slideshow. And I hope you can see that. Okay, can you see the screen? Yeah. I'm going to tell you about some of our recent work in which we've been looking at.
Hello, sorry to stop you. The screen is actually not visible.
Hold on.
As in your slides are not visible yet.
Oh, hold on.
May take a couple of seconds.
Yeah.
Because it says she started screen sharing, but sometimes it's a bit of
some kind of lag maybe.
Shall I stop sharing and try again?
Yes, that would probably be the best, Fiona.
Yeah, I'll stop. Okay, now you can see me.
Yes.
Try and share. How's that? Can you see that?
Yes.
Perfect.
You can see the shard.
Okay. The shard. Well, I like to start with this picture which shows my lab. My lab isn't in the shard, but we're on the top floor of Guy's Hospital. It's afternoon in UK, although it's evening in India, but we have a nice view at all times. Anyway, I'm going to tell you today about a long-standing interest of mine, which is in heterogeneity of cells in human epidermis. And I'm going to give you a little bit of a historical perspective on that. Then I'm going to tell you about some of our cell-based in vitro experiments. And then I'm going to ask how new data sets involving single-cell RNA sequencing of cells directly isolated from the skin, how that is validating our older findings and actually giving us ideas for new mechanistic experiments to carry out. So for those of you who are not familiar with the epidermis, it is the outer covering of the skin. And I'm going to, for the purposes of this talk, really focus on what we call the interfollicular epidermis. So I'm not going to talk about hair follicles or sweat glands or anything like that. And really just think about the epidermis as a beautifully simple tissue, which is made up of multiple layers of epithelial cells called keratinocytes, and they're separated from the underlying connective tissue, the dermis, by a specialized extracellular matrix, which is called the basement membrane. And you can, in this histology side, you can just make it out as this sort of pink area here. Now, anybody looking at this section of epidermis would realize that the cells in the deepest layer, the layer furthest from the surface of the skin, have got nuclei, but the cells out here don't. And in fact, this structure of the skin is one of the reasons why people realized many, many years ago that it must be maintained by stem cells. And what we know is that cells in the basal layer attached to the basement membrane divide, and they give rise to cells which move out of the basal layer, stop dividing, and then undergo a process which we call terminal differentiation as they move through the upper layers into these outermost or cornified cells. These outermost cells are dead cells. They don't have a nucleus or any cytoplasmic organelles, but they're very important for our lives because they act as a barrier to protect the skin from invasion by pathogens and regulate hydration of the skin. So we've been fascinated for a long time in what is the nature of the stem cells in the basal layer and what determines how they exit the stem cell compartment and produce these differentiated cell layers. We know a huge amount about the different layers of the epidermis. We've got lots of markers of the different cell layers, and over the years, we've come to find out about some of the different properties that they have. But I'm going to focus, first of all, on the stem cell compartment. So the cells which are ultimately responsible for maintaining the tissue. And as you heard, I was a postdoc at MIT with the late Howard Green. And Howard, back in 1975, with his student Jim Reinwald, published the observation that you could take cells from healthy human epidermis and plate them at single cell density on a feeder layer of 3T3 cells. And when you did that, some of the cells would start to divide and they would form little colonies of cells. But within the colonies, you'd start to see multi-layers forming. And in fact, in the dish, they were recreating that simple structure of proliferation in the basal layer, differentiation in the suprabasal layers. Now, although Howard never practiced medicine, he did train as an MD in Canada. And I think that is one reason why, as soon as he had made this discovery about the cell culture model, he realized that it could potentially be applied in the clinic. And only 6 years after the first paper describing the cell culture method, he, with colleagues, showed how it was possible to take a biopsy of skin from a man who had suffered a burn on his arm, expand the cells in culture, and graft them back onto the patient. And so this technique, which is now many years old, has continued to be refined and pursued in other epithelia like the cornea and combined with gene therapy. So it's a classic example of a stem cell therapy which involves in vitro expansion of the cells. So we know that the stem cells persist in culture because otherwise the grafts wouldn't work. But we needed to have some ideas about how we could identify and count stem cells. And my lab and several others focused on this ability of some of the cells to form colonies. And there are different ways of doing this assay, but we use colony formation as a quantitative measure of human epidermal stem cells in culture. And this just shows you an experiment in which we were testing different batches of culture medium to find which ones were best. We remove the feeders and we stain the keratinocytes with a pink dye. And you can see very easily that there are fewer colonies here than here, and this here than there. And so it gives us a readout for assays in which we're trying to enrich for stem cells. Of course, looking at colonies in a dish is not the same as looking at skin as a whole, but there are classic techniques in which you can reconstitute human skin in culture, and that's just illustrated here. So what we take is a piece of surgical waste skin. We remove the epidermis and put the cells in culture. Then we kill all of the cells in the dermis by repeated cycles of irradiation and free thawing. And then we place this decellularized dermis on an insert well. And then what we can do is inject fibroblasts back into the dermis. You can see a few fibroblasts in that section there. And then we plate the epidermal cells, the keratinocytes, on top. And when the epidermis and we feed from below, when the epidermis is reformed, we get this nice dry surface appearance compared to if we don't add any keratinocytes. And then when we cut sections through this, we get a reasonable reconstitution of the skin. So in many of the experiments that we do, this is our test for how good the reconstitution of the epidermis is. Now, we've been looking for stem cell markers for many years. And initially what we did was to take antibodies to proteins on the surface of basal cells and ask whether we could flow sort clonogenic cells with antibodies to these candidate markers. And the first marker that we found, which turns out to be a classic marker that's important in many tissues for enriching for stem cells, is that we found that our clonogenic cells expressed high levels of beta-1 integrins. So these are extracellular matrix receptors. And we also found that a quick way to enrich for the stem cells was to plate keratinocytes on extracellular matrix like fibronectin or collagen and select the cells that adhered most rapidly. So the cells with the highest levels of receptors adhere most rapidly to the extracellular matrix and are enriched for clonogenic cells. And we found quite early on that although all cells in the basal layer express integrins, if we looked at the cells with the highest levels, they weren't randomly distributed, but they were clustered within the basal layer of the epidermis. And over the years, many labs have looked at the signaling pathways which are downstream of the integrins. So the integrins are actually heterodimers of an alpha and a beta subunit. They bind the extracellular matrix, and you can see many interacting partners. And over the years, many different functions have been ascribed to the integrins. And I'd just like to draw your attention to the fact that in the epidermis, one of the key pathways downstream of the integrins is ERK-MAP kinase. And if you reduce integrin-mediated adhesion, you can rescue colony formation by overexpressing a constitutively active mutant that activates ERK-MAP kinase. The other candidate marker that I want to describe was again one that we found early on, but this was actually by talking to our neighbors in a Drosophila lab who were very excited that the Notch pathway, which is a classic Drosophila signaling pathway, turned out to have mammalian homologs. And we found that in human epidermis, we could see evidence that clusters of cells in the basal layers expressed high levels of the Notch ligand delta-like 1. So this shows you in this review of Notch signaling how the delta and other Notch ligands are actually anchored in the plasma membrane, and they're signaling to Notch on neighboring cells. And over the years, relying largely on overexpression studies, we made some interesting observations which suggests that expression of this ligand, DLL1, by stem cells stimulates neighboring cells to differentiate. So here stimulates neighboring cells to differentiate and also mediates the clustering of the cells independently of Notch signaling. So here we've got another cell surface marker that enrich for stem cells. And what it's doing is, we believe, instructing neighboring cells how to behave. So the conclusions from our studies of cultured human epidermis are that there are multiple populations of keratinocytes in the basal layer, stem cells and non-stem cells. And we've got some markers which allow us to enrich for stem cells, and we know a little bit about the downstream signaling pathways. Another population of cells in the basal layer that we've been very interested in are the cells which are described as being committed to differentiate. So these are cells which are no longer stem cells. They haven't yet left the basal layer and begun to differentiate, but we know that at some point in time, they will differentiate. And for a long time, it was difficult for us to find out about commitment because we didn't have any markers to enrich for committed cells. But a few years back, we were able to find those markers by making use of another classic Howard Green experiment. And this is one in which Howard was growing cells in keratinocytes in culture. And he found if he disaggregated them and put them into suspension in methylcellulose supplemented medium, the cells couldn't attach to the substrate. And if he followed them for 3 or 4 days, he found that they turned into cornified cells from the outermost layer of the epidermis. And in work that my former postdoc, Joe Adams, and I published a long time ago, we obtained evidence that the point at which cells in suspension decide to differentiate is 4 hours. Now, we, of course, use beta-1 integrins as our marker for cells that are in the basal layer. And the marker for differentiation that we tend to use is this protein involucrin. So at 0 hours, we are enriching for integrin-positive, involucrin-negative cells. at 24 hours, the cells have downregulated their integrins and switched on involucrin. This is qPCR for a couple of basal markers. You can see between 0 and 4 hours, levels are high. You can see involucrin and another differentiation level marker are low at 4 hours and increase later. But what we're arguing is that at this 4-hour time point, something is happening which is switching cells out of the stem cell compartment. To find out what those events might be, we had a wonderful collaboration with Tony Lee and Angus Lamond in Dundee, who are experts in proteomics. And what we did was simply collect cells at different times in suspension. We carried out colony forming assays to make sure that the cells were leaving the stem cell compartment. We carried out transcriptomics. Tony and Angus SILAC labeled the cells and did proteomics. And this allowed us to look at what was happening at this commitment point. About 18 months ago, my postdoc, Matteo Vietri-Rudan, published another data set in which we carried out a lipidomic analysis. I'm not going to tell you what we found when we looked at the lipids in the cells, but if you're interested in integrating protein, RNA, and lipid data, I think the two data sets are well worth exploring. So what did we find at this 4-hour time point? Well, we found that at this commitment point, there was a transient upregulation of quite a large number of protein phosphatases. Just to illustrate this, this is qPCR showing the decrease in a basal cell marker, P63, which goes down after 4 hours. And here are examples of 3 different protein phosphatases, which you can see peak at 4 hours and go down after that. We wanted to know if there was any evidence that they were regulating colony formation. And so we knocked them down individually, and then we quantitated colony formation. And in this spider's web diagram, each circle represents 10% of the cells being able to form colonies in culture. And our scrambled control here in red is just over 20% of the cells. And then for the individual knockdowns, you can see what effects we saw. So there were some, like this one here, where knocking the phosphatase down decreased colony formation. But we were particularly interested in ones like this one, PTPM13, DUSP6, where the knockdown increases colony formation. And we argued that this meant that the upregulation of these phosphatases would normally drive cells out of the stem cell compartment and promote commitment. Now, we published the data a few years ago, so I'm not going to take you through all of the arguments that that conclusion was correct. But I will show you a couple of the interesting things about the phosphatases. Although we'd found them in a proteomic screen, we discovered that they were very tightly regulated at the RNA level. And we also found that if we knock down expression of one phosphatase, it would affect expression of the others. So this is a heat map showing mRNA levels. And just to illustrate, if we knock down this phosphatase DUSP10, we get upregulation of these 5 phosphatases over here. And conversely, if we knock down DUSP6, you can see that we get an upregulation of DUSP10. So we were really puzzled to understand what was going on here. And we were fortunate to be able to collaborate with a bioinformatician called Sarah Jane Dunn, who took all of our RNA and protein data and modeled the different levels using Boolean network analysis. So essentially for a biologist like me, that means taking the gene expression levels and abstracting them as either on or off, depending on whether expression went up or down. And these show you the interactions at time 0, at 4 hours in suspension, 8 and 12 hours. And what was interesting was that in the stem cell compartment and in the differentiated cells, many of the interactions were negative interactions, as you can see by these red T-bars. But at the commitment point, you can see lots and lots of positive interactions. And this suggested to us that this commitment point is unstable and that the protein phosphatases are acting as an unstable switch between two stable states. So the model that we have, which is shown here, is, as I've said, that the stem cell state is stable. When we trigger cells to differentiate, they become an unstable state through this network of protein phosphatases. And then once that switch is thrown, the cells move to a stable differentiated state. Now we know a lot, I told you about the signaling pathways downstream of integrins and other extrinsic signals. And it was interesting, I highlighted the importance of ERK-MAP kinase signaling. When we looked at the top 15 dephosphorylated peptides in our data set at commitment, 3 of them turned out to be ERK-MAP kinases. So that would argue that at commitment, there's dephosphorylation of ERK. And we also know that AP1 transcription factors are very important in regulating many of the differentiation genes. And sure enough, we found that some of these were upregulated in suspension. These are 2 examples here. And then if we knock down the phosphatases, that reduces the levels of the AP1 factors. So more recently, what we wanted to do was start looking at this commitment switch at the level of individual living cells. And to do that, we were able to work with Michiyuki Matsuda in Kyoto. We were fortunate that his PhD student, Toru Hiratsuka, had moved to my lab as a postdoc and brought tremendous expertise in FRET sensors and FRET signaling to the lab. So Toru used this ERK-MAP kinase FRET sensor, which is very nice because it reports ERK-MAP kinase activity in the nucleus. And so we used a retroviral vector to introduce the sensor into our cultured human keratinocytes, and we combined it with a fluorescent reporter for the differentiation marker involucrin. So now what we could do is in individual cells, look at ERK activity and correlate it with the upregulation of a differentiation marker here. So this slide shows you 4 individual cells, which we're monitoring for a period of 12 to 16 hours. And in each case, you can see that over time, the red signal increases because the cells switch on involucrin. And what we noticed immediately was that before differentiation occurs, there's a period of pulsatile ERK activity, and then subsequently ERK levels go down, which is what we would have predicted. So this got us very interested in whether a pulsatile ERK activity was indicative of commitment to differentiation. We also wanted to know what were the effects of DASP6, of protein phosphatases on ERK levels. So this slide just shows you the effect of overexpressing 2 of the protein phosphatases, DASP6 and DASP10, on ERK activity. Now, in the case of DASP6, when we switch it on, we don't see any effect on mean ERK activity, but we see ERK pulse levels decrease. And conversely, with the other protein phosphatase, DASP10, we see that mean ERK levels go down, but the pulses stay the same. So we published this work 18 months ago. And just to summarize what we think is going on here, we believe that we've got a stable stem cell state with high ERK levels and no involucrin expression. And when the cells have differentiated, involucrin levels go up and ERK levels are low. And we've been able to distinguish 2 different pulsatile ERK states, one in which the pulses are happening and mean levels are high. And when cells are in this state, they can return to the stem cell compartment. But if the pulses occur and mean levels are low, then those cells will irreversibly differentiate. So we believe that the different protein phosphatases are exquisitely tuning ERK-MAP kinase levels to drive exit from the stem cell compartment. So to summarize what I've said so far, we've just as we used a candidate approach to look for stem cell markers, and what we did to look for markers of commitment was to take a time course approach, which led us to identify a network of interacting protein phosphatases, which impact on ERK MAP kinase activity. And I didn't show it to you, but in vivo imaging of mouse epidermis also reveals a patterning of pulsatile ERK activity. So what has this got to do with real life? We all love our experimental models. We put a lot of effort into them. But from time to time, we need to check whether what we've seen in our experimental models maps onto the situation in vivo. And this is why back in 2016, I was very excited when an initiative called the Human Cell Atlas was launched. This is a global initiative which has the aim of mapping all of the cells in the human body. It's like Google Maps for the body. And the intention is that this will give us new insights into how the body works and what goes wrong in disease. But I think equally important, it encourages collaboration and data sharing, and it helps us to integrate different types of data sets. Now, I had a personal reason for being very excited about this initiative, and that was that back in 2006, with my then postdoc, Kim Jensen, we had tried to do single-cell gene expression profiling on our cultured keratinocytes because we wanted to find new stem cell markers without relying on a candidate approach. So what we ended up with was about 300 cells from basal layer of cultured epidermis. We could distinguish the differentiated cells and discard them. And then with the cells which hadn't yet begun to differentiate, we could look for cells that expressed a couple of our stem cell markers, including DLL1. So we only had 9 informative cells, and the level of confidence in differences in gene expression was 7-fold, which of course is massive. Nevertheless, this was useful. It led to a new marker of epidermal stem cells, which Kim went on to show is hugely important in a variety of epithelia and other tissues. So when the Human Cell Atlas was launched, we were very fortunate to be able to get funding from the Wellcome Trust on a project which is led by Mazlifa Hanifa and Sarah Teichmann to carry out single-cell RNA sequencing on a scale which would literally have been unbelievable to us 20 years ago. And the team at King's who've been working on this project is led by Magnus Lynch. So instead of working exclusively with cultured keratinocytes, all of the cells that we're analyzing have come directly from human skin. And we've compared epidermis with dermis, immune cells, and so on. And we've got data for healthy adult skin, developing skin, so fetal skin, and also for 2 inflammatory diseases, eczema and psoriasis. And the total number of cells that we've been able to analyze is over 500,000. And as new data sets are published and shared online all of the time, the true number of cells that we can analyze now is probably closer to 1 million. So what does this tell us about what can this tell us about our in vitro experiments? Well, we predict from our experiments with cultured epidermis that there are multiple populations of cells in the basal layer, that transient upregulation of protein phosphatases triggers the start of differentiation. And we believe that components of the Notch pathway are differentially expressed in basal cells and differentiated cells, and in particular that DLL1 is expressed in the basal layer. So what we've done most recently, and this is the work of Victor Negri, who's left the lab recently to go and work with AstraZeneca, is to take the published data set and exclusively look at epidermal cells from the healthy samples. And using UMAP plotting, he was able to identify 13 different cell states. Now, because we have so many markers of the different layers of the epidermis, we can be completely confident that this set of markers, this set of cells all belong in the basal layer, whereas these ones belong in the differentiating cell layers. So the question is, if we focus here in the basal layer, what are the markers and do they relate to what we'd seen previously? Well, if we look at basal 1, 2, and 3, as they're called, we could see differential expression of markers that we and others had identified in the stem cell compartment. Interestingly, beta-1 integrins was most highly regulated in basal 3, DLL1, which is expressed at tiny amounts in basal 2, together with other markers like CD46. And then basal 1 was enriched in 2 markers that we've studied a little bit called caveolin 1 and 2. And of course, we can use lineage inference techniques like slingshot to try to predict the relationships between the different populations of cells. And the questions that we're focused in on at the moment are whether these different populations of cells are functionally distinct, or whether, in fact, the different levels of RNA are not so important because, of course, the half-life of the proteins will be different. And we also want to understand whether the functional relationships between those populations of cells. Now, what about the exit for the commitment genes? Well, here are the qPCRs that I showed you before, and you'll notice that the peak for DUSP6 and DUSP4 is sharper than for DUSP10. So interestingly enough, in Victor's UMAP plots, we find 2 transition populations, transitions 1 and 2. And if we look at them, we see DUSP6 and DUSP10 in those populations. But you can see that the distribution of DUSP10 is broader than the distribution of DUSP6. So we were really pleased about that as well. Now, when it comes to the notch pathway, there are so many genes, it's really is worth a talk in itself. But we've mapped the different receptors and ligands to different subpopulations of cells. As I showed you before, DLL1 shows up in basal 2, as do 2 other ligands, JAGID1 and JAGID2. And it's been known that although DLL1 promotes stemness, JAGID1 and JAGID2 stimulate cells to differentiate. And consistent with that, we see the upregulation of the receptors in the transition populations and in the more differentiating cell layers. So it's a complicated pattern, but it fits quite well with the published biology. So the last question I want to ask in my presentation is, we can pat ourselves on the back. We can say, yes, we've been able to validate the experimental models using single-cell RNA sequencing. But to make it really useful, we should be able to go back into the data set and design new experiments. And I'm just going to end with one example of what we've been able to do. And that has been ask the question, why is it that DLL1 is expressed in such low levels and yet has such powerful effects? Could there be a mechanism that would explain how the effects of DLL1 are amplified? Well, our attention turned to a set of genes called the Fringe genes, because they are known to add carbohydrate residues to the EGF repeats of the extracellular domain of Notch. And this is known to alter the affinity of Notch for the different ligands. So different Fringe genes will increase the affinity of Notch for DLL1 versus JAGID. So what we could do is go into the single-cell RNA-seq data set. When we did that, there are 3 Fringe genes, manic, radical, and lunatic. And we found that lunatic Fringe was more highly expressed than radical Fringe, and we couldn't detect manic. To validate that, we isolated RNA from epidermis of 5 healthy samples, carried out qPCR analysis, and this confirmed the single cell RNA sequencing, so that lunatic fringe is by far the most abundant fringe gene. And then in culture, the same is true, although we do see relatively more upregulation of manic fringe, and the difference between radical fringe and lunatic fringe is less pronounced than we see in vivo. But nevertheless, this is really intriguing because lunatic fringe increases the notch affinity for delta-like 1, and it can also inhibit cells from responding to Jagged. So the question is, if we look at the effect of lunatic fringe in our cultures, can we see any evidence that it promotes maintenance of the stem cell compartment? So this shows you some of our most recent experiments. We've confirmed that we can overexpress lunatic fringe using a retroviral vector. I won't take you through all of the controls, but I will point out that by qPCR, cells overexpressing lunatic fringe show a downregulation of involucrin and other differentiation markers. But the effect, which I think is really striking, is if we do the colony formation assays, we can see that overexpressing lunatic fringe gives us a big increase in the number and the size of the colonies, arguing that it is indeed able to amplify the effects of delta in the stem cell compartment. So the model that we have of how delta acts in the basal layer of the epidermis is that it's expressed in the stem cell compartment. It stimulates neighboring cells to differentiate. It protects stem cells from differentiating. And these effects are amplified by the effect of lunatic fringe. So I'm almost at the end of the presentation. I just wanted to give you a sort of a snapshot of where we're going next in our experiments. And for a long time, we've been interested in analyzing interactions between stem cells and their local environment in a dish at single cell resolution. So we've done a lot of experiments where we've plated single cells on adhesive islands of different areas and looked at how differentiation is affected. But for a long time, we've really wanted to look at the effects of neighboring cells. And this has been quite frustrating because by and large, we've not seen any effects. But what I want to end with is some work that my student Blaise Louis has done very recently. And it's a negative result and a positive result. So she found that she could engineer cell-cell interactions in suspension by using lipid-modified oligonucleotides to bring cells together. So you put one DNA strand into the membrane of one population of cells, the complementary strand into its neighboring cell, and this will give you nice aggregates in suspension, as you can see here by flow sorting. And I should say this is a wonderful collaboration with Zev Gartner and David Schaefer in San Francisco. So sadly for us, making these cell aggregates in suspension had no effect at all on the protein phosphatase network or on differentiation. But we did see some positive effects when we engineered cell-cell interactions on micropattern substrates. And the way we do this is to make islands coated with extracellular matrix that are shaped as hexagons. And we find that if we plate cells on large hexagons of 44 or 55 micron diameter, the cells do not differentiate, but on the smaller hexagons, they do. And interestingly enough, if we found that if we look at these bow tie islands, we see situations in which neither cell has differentiated, but we never see a situation in which both cells have differentiated. If there's differentiation, it's always happening preferentially in one cell and not in another. Now, as far as we can see, the cells that we're plating are the same and the microenvironment is the same. So we dearly want to be able to look in future at what might be intrinsic or extrinsic that is causing this asymmetry of fate. But that will be a talk for another time. So to summarize, the single cell RNA sequencing work that we've done has confirmed the existence of multiple, that should be epidermal cell populations. It's a reality check for our experimental models, and it is giving us much more information about how the skin works in health and disease. And it has been great fun working with computational scientists, people who are experts in imaging, to pull the data together. So with that, I'd like to say a huge thank you to my lab. This was July. We had thought we would be able to have a party to celebrate the end of COVID, how wrong we were, but we were allowed to meet outside for a game of rounders, and we really enjoyed that. I think we spent most of the time talking rather than playing, but I'd like to say a huge thank you to them, and also a huge thank you to you for listening. So I'll stop sharing and I'd be happy to take questions.
Thank you, Fiona, for this wonderful talk. I love the way you went all the way from the sort of early aspects of suspension culture, all the way through to single cell transcriptomics and back to suspension culture, which was also very exciting. So while I wait for people to post their questions in the chat box, I'm going to ask you, what do you think, it's an old question, but what do you think is the difference between, responsible for the difference between the way in which mesenchymal cells and epithelia respond to lack of adhesion? Is it related in any way to their developmental origins, or is it, just a cell surface wiring problem, mechanochemical wiring?
I mean, it's interesting that if you put normal fibroblasts into suspension, they will stop dividing and they'll shut off RNA and protein synthesis. That was old work, I think, from Sheldon Penman, who was next to Howard Green back in the day at MIT. But I think it's important to bear in mind that in an epithelium, it will be polarized because only one surface will be in contact with the extracellular matrix, whereas fibroblasts are intimately linked on all surfaces to the ECM. And we've done a lot of studies on fibroblasts, particularly in mouse skin, and we've been really surprised at how quiescent they are when they're associated with ECM. So it's a fibroblast in a dish is not much like a fibroblast surrounded by ECM.
Yeah, that's interesting. So, okay, we have a question from Abhishek, who says, is there any baseline for controlling the cancerous stemness in heterogeneity of cell differentiation?
The topic of cancer stem cells has been around now probably for 15 or 20 years. And I suppose at the time when it began, for me working on squamous cell carcinomas, it seemed kind of obvious that the cells which were going to divide and maintain the cancer were the undifferentiated cells. But I think things have moved on a lot since then. And you can see that in types of cancers we study, the cells that aren't dividing can still have a profound effect on tumor growth because they're communicating with the cancer stem cells all the time. Now, what the experiment that's cooking at the moment is one in which we've got single cell RNA-seq data for human basal cell carcinomas. So I can't wait to find out if we can see our three populations of basal cells in there, if they're completely different or if one is dominant over another. And the great thing about this analysis is that we'll also get information about the fibroblasts. So we can say, what actually is a cancer-associated fibroblast?
Yes.
So don't have the results yet, but it's very, very interesting. And the great thing about these data sets is once you learn to analyze them, they're completely freely available to anybody. So you can download them, you can play with them, you can intersect data from different labs. It's very exciting.
Yes. Yeah. One of the things that I was, looking at your UMAP sort of schemes of your single cells, and I was wondering if you've seen cell populations which sort of overlap in the transcriptomic space in some aspects of the genes that they express. Are there clusters of genes which allow you to now define new populations which you can then say have different phenotypic consequences?
It's a good question, and it really actually makes me declare that the trouble with making UMAP plots is that what you get depends on what you put in.
Yes.
And so the more data you pile in, the more populations of cells that you see.
Yeah.
And what we've seen in our most recent work on the fibroblasts is that by just putting tons of data in, we can start to see new populations of cells, which just they're not sufficiently abundant that you see them if you don't have a big sample size. And the other thing is that there is a distorting effect. For example, in the basal layer of the epidermis, proliferating cells show up as a separate cluster because they express cell cycle genes. Yes. But actually they correspond to a subset of the other basal population. So I think one thing I've learned is not to get too excited and run off and do an experiment. Just wait, because the clusters can change. Yeah.
That's absolutely true. So okay, Kaushik here has a question about how can one explain that the same set of phosphatases, which are negatively regulating each other at 0 hours in the stem cell phase, turn to positive regulation at 4 hours in the committed phase?
That's a really good question, and one that I can't answer properly. The one thing I would say is that if you look at transcript levels at single cell level resolution using RNA FISH, you can see there's a really tight positive correlation between levels of ERK transcripts and levels of DUSP6 or DUSP10 transcripts. So, and it's published by other groups that there's a very short half-life of the transcripts, and they're being regulated at this transcriptional level as well as post-translationally. So you could, I'm hand waving literally, but you can invoke post-translational effects on the proteins and the fact that the phosphatases have multiple targets, plus these transcriptional effects. I personally believe that these networks of protein phosphatases are used again and again in lots and lots of different cell types.
Yes. So, actually I had a sort of related question of that. You showed that the DUSP pulsatility was happening at different levels and selecting different targets potentially, right?
Yeah.
So what happens if you just, you retain the high level or the low level, but you inhibit the pulsatility by sort of holding them in one state or the other? Flip cell fate?
If you treat with a pharmacological inhibitor of the pathway, a MEK inhibitor, you can go straight from stem cell to differentiated cell and bypass the pulses. So they're not compulsory. Yes. And if you monitor a single cell in a pulsatile, high pulsatile state, we had this one champion cell which was pulsing for hours. And then in the end, it divided. And the two daughters went to the stable high state. I think the way to interrogate this more is using optogenetic tools which exist.
Yes.
And I would also really like to dial in a reporter for a P38 MAP kinase, because if you activate that, the cells will differentiate.
Right.
But it's
be a fun experiment.
It's fascinating. Yeah.
Okay, Purna has a question which says, do you think that there is lateral inhibition between stem cells grown on smaller hexagons of the biomaterial substrate? And could that be delta jagged mediated?
I think so, yes. We haven't done the experiment. One of the things that intrigues me is that when you label cells, if you overexpress DLL1 and label for delta like 1, you see lots and lots of endocytic activity. And caveolin 1 and 2 are definitely upregulated in some of our cells. So in my mind, I imagine the two cells sitting together, if you like, communicating by reciprocal endocytosis. And then at some point, one cell's got more delta, and that means tough, this cell's got to differentiate. But that is total speculation. We've got to do the experiments.
Yeah. Okay, here's a question from Venkat's lab, but I don't know who it is. It is, how big the challenge would it be to analyze human cells of global populations with respect to the Human Cell Atlas, considering the ethnic and regional variation across the globe? So there's literally global, not just the human cell population, but the human population.
It is a great question. It hasn't been done systematically, but there are definitely, I know there's really good data for the Han Chinese population. Our samples are white Northern European. This isn't very global, but I know that our samples are totally overlap with whatever was sampled in California. So
no sulfur effect.
So I would speculate that what you might see is differences in relative abundance, but I could be wrong. I don't think you'll see any different cell types. And in fact, we know if you look at melanocytes, for example, which give the skin its pigment, they differ between people of different skin colors by the number of pigment granules, not by the number of the cells or their nature. So yes, it's interesting. And I must say that the first time I shared my data with somebody in America, we had it on our laptops, and I thought this is going to be a disaster because it's going to say that cells in London are different from Philadelphia, but the mapping was, our jaws dropped. So I think we're more alike than we might sometimes think.
Well, that's a lovely unifying message. So I'm going to ask you a really off-the-wall question to end, because I don't see any more questions. In your micro patterns, you saw these asymmetric fate choices that were happening. Do you have any evidence for the immortal strand in your micro patterned dishes?
Yes, so the immortal strand hypothesis, I don't know if your students know, was that to be a stem cell, you keep the immortal strand of your DNA, and you share the newly synthesized strand with other cells. I haven't thought about this for quite a few years, but I don't think that the labeling experiments that have been done, at least in the mouse, are consistent with that hypothesis. It was a great hypothesis, but the evidence for it at the moment is not great. It served its purpose.
Yes. So if there are no further questions, we'll thank Fiona for a fantastically engaged evening and really giving us food for thought, and for spending time with us. And thank you to the CCMB students for organizing this. And I just have to say one thing, because I just got a text message from the place where I now work, which is the India Alliance. And some of the scientists said, you didn't give a plug for the India Alliance. Fiona Watt was our chair of the Early Career Fellowship Committee for several years ago, and everybody was delighted to be able to see you online because they loved having you as the chair. So thank you, Fiona, both from CCMB.
Yes.
And
let me say that the India Alliance it's fantastic. I miss my colleagues there so much.
Oh, good. Maybe we can have you back.
I would love it. That's how I've been to Hyderabad, is through the India Alliance.
We'll have you back as soon as the COVID lets us. Thank you then.
Thank you all, and enjoy your
all
evening.
The best.
Yes.
Group. Yes.
Take care.
Bye-bye.
Bye-bye.